Welcome to Let's Talk, a place for straight talk, hard topics, and smart people. I'm Sue Wallace, and I'm here today with Rainier Hernandez, and really excited to have you join us. Thanks, Rainier, for being here.
Thanks for the invitation. This is a lovely opportunity to get this conversation going and hopefully add something to the discourse of the community.
Absolutely. So I have to say this as an introduction for everyone that is going to be joining us later, Rainier, to watch this video. I've hosted a lot of Let's Talk conversations. This is one of my favorites. I'm going to be one of my favorites. And not everyone knows that, may not realize I'm actually a radiochemist in the beginning. And I'm so excited to have this chance to sit down and pick one of the greatest theranostic radiochemist brains here. So I'm going to learn something, and it's going to be
I'm honored for all the credentials. But I mean, I'm very happy to just provide my 2 cents to the discourse here.
So tell us, just start by telling us a little bit about, I mean, you're doing a lot of things. You're a physicist, PhD physicist, you're a, radiochemist, buried in the middle of developing all these novel theranostic agents. What are you actually doing right now? What's your focus? Because you can do it all.
Like you're saying, that I have 2 main focus, and I'm essentially that the overarching focus is just how do we make these therapies, that potentially life-saving therapies, better? How do we make them, optimize them? And there's a few ways that we can do that. And that kind of my lab is divided in two main veins. Essentially, the agent development things, just try to generate or create better agents that have better efficacy, but also on the radiobiology side of things, because that's kind of the yin and yang here. So we sometimes we get too focused on the agents, but we forget about there's things that we can do on the radiobiology side of things to make these agents more effective as well.
So what's your hit list then? What are the things you think about? Is that I need to come up with a new drug? How do you organize your thoughts?
I think, like you're saying, my background, I did an undergrad in radiochemistry, then a PhD in medical physics. So I understand, or I think I understand fairly well, how the mode of action of this radiopharmaceuticals, which is in the end radiation. So if you understand radiation, that is kind of like your yardstick. Okay? We give more radiation, we get better efficacy. We know this from external being, but it's not self-evident that that's the way radiopharmaceuticals work. Because most people think of this as drugs, because of the way we administer them. But in reality, this is a radiation therapy modality, right? So, and a lot of the rules of engagement that we know from external being radiation, for example, apply to radiopharmaceuticals.
So help me understand what you just said. Rules of engagement in what regard?
We know that for example, loyalty radiation will kill through DNA damage, reactive oxygen species. That's common between the radiation modalities. I mean, for example, brachytherapy. If we were to draw a better parallel between the conventional or historical radiation therapies, brachytherapy is very much like a radiopharmaceutical therapy when you have a low dose rate and those considerations. Say a lot of the same predictors of response. We know now with all the advancements in the last 10 years in radiopharmaceuticals and the symmetry, we know we're starting to learn what are the absorbed dose levels that we need to hit in order to elicit efficacy. So there's some evidence already there, but I ground a lot of my work on the symmetry. Say, okay, how do we make agents that improve the dosimetric distribution in the patient? So and there's a few ways that we can do to try to maximize that.
So when you say that, let's optimize the distribution of this drug, are you just thinking about, obviously you want it in the tumor or on the tumor and not in advance? Do you generally know what the critical organs might be for this sort of a molecule? So you sort of know the areas you're worried about, and that's what you're trying to balance.
And that's that's the other beauty of this field, is that it's one, I would say the only example where you can see where the drug goes. So no other drug modality really can do this effectively, right? So we can predict not only the distribution, but the dosimetry too. So so I can, with very effectively and safely, gauge whether these patients are gonna benefit and whether we're gonna be hitting the marks that we need to get efficacy.
So we like at Voximetry, we have a very interesting perspective on your not your personal work, but the radiochemistry work that's being done out there on all these
Yeah.
molecules. Because as you know, they come to us later in a clinical trial and they say, "Here's the images we have, and you help us look at the dosimetry here." And then they say, "What does this tell us about what's happening in this patient?" And all we can say is, "Well, we can see there's a radionuclide here."
Mhm.
And I'm always thinking, I'm wondering what happened to that molecule. I mean, how do you have a way to know if these things are staying, bound to the ligand? Do you care? When does it matter? How does that factor in?
I mean, it's this. You can't just open the Pandora's box here. So it's
You
a
can
lot
handle
of,
it. You can handle it.
there's a lot of moving parts in that question. So okay, if we're thinking about like 1 decay, like classical beta emitters, we deliver it there. It decays, not a problem. So what you're mentioning, it factors in, or is more of a consideration when you're looking at decay chains and the possible redistribution of daughters, and yeah, everybody wants to know where the daughters are. I want to know where my
Yeah.
daughter is
Yeah,
right now.
especially at midnight. Especially at
But I
midnight.
mean, but professionally too. So
Yeah.
and like I mean, actinium. Obviously, there's a lot of interest in actinium because of that's one of the ways that, as I was mentioning, to make the agents better. For example, I mean, just based on energy, I always try to like explain this more to the chemistry audience, right? Say, okay, we have lutetium. Lutetium will give you about 0.5 MeV of energy release per decay per atom, right? And you look at actinium, that and you sum all the energy decay, all the energy deposited by the whole decay chain, we're talking about 30 or more MeV. So we're looking at a 60x more energy per atom
Yeah.
of actinium. Actinium has 60 times more energy to give compared to lutetium. So even if you don't change anything in the agent, you just swap the isotope. Now you got a drug that is potentially 60 times more effective, right? And there's other considerations about like linear energy transfer and microdosimetry that also play a big factor on the potential upside of the alpha emitters. But now, because they're more potent, they could be potentially more toxic too. So
Yeah.
now it becomes really important to know where all the decays are happening, right? So, and obviously, it's a lot of interest in the field, like tracking those daughters and see how they redistribute. So this is like, I think it's a 5-year-old problem. So we're still very early stages,
Yeah.
and we're still figuring out technologically how we can tackle this. But
So you've got a lot of options when you think about how to approach a new. You just think about a new radiopharmaceutical that might be useful for everyone. How do you approach that? What are your options?
so
The factors.
the first thing that we go is try to identify the targets, and that turns out to be the biggest problem, or the biggest barrier to radiopharmaceutical development. Finding targets that have some features that are amenable to be targeted with
You're
radiopharmaceuticals.
talking about a molecular level target,
At the molecular
something
level,
can stick to.
the cancer,
Yeah.
exactly.
Yeah.
I mean, and like I say, different modalities have different boxes that you need to hit for a target to be good.
Such as.
Yeah, if to say, okay, overexpression of the target in the tumor cells versus the other cells. For us, we need that the targets are expressed in general in the membrane, because that's what the radiopharmaceutical seeks, right? So that's how we achieve selectivity. So there are other treatment modalities, cancer treatment modalities like TKI and things like that they achieve selectivity on the biological pathway that they inhibit, right? But for us, wherever we put radiation, we deliver radiation will have an effect. So for us, the therapeutic index is on this delivery. It's essentially a drug delivery issue.
Yeah. But it's really interesting because, sort of the training that a radiochemist goes through and in their first 2 years of postdoc work or whatever it is, they're not taught anything about radiobiology. And you're telling me you need a lot of expertise in radiobiology
Yes.
before you get to start your radiochemistry work.
And that's not unfortunately, but that this is the aspect that I feel is most behind in the field. So we have exceptional radiochemists. We understand how to make these agents. We understand the physics very well. There's like a lot of effort into working on the supply chain, so we have access to like high quality isotopes. But the radiobiology was still we're not really quite understanding what. For example, many of the Pluvicto patients don't respond, even though they have like good uptake.
Yeah.
So why some of the lesions don't take the agent, other lesions
You're
take
seeing
it?
uptake, but still no response.
Yeah, I mean,
So-
like even in
So
like,
they're radiation resistant, perhaps.
Yeah.
Right. Something.
and we're starting to understand that DNA repair, the radiosensitivity context, is a big deal. I mean, there's a study, very elegant, a study that for I think it's from at least a day, just being involved in that, where they essentially came up with a biomarker to predict response to Lutathera. So it's called a PPQ, at essentially it's blood based and then some histology biomarker, and then you can pick the responders with a 95% sensitivity. It's amazing but it's a more biology based biomarker.
Yeah.
So things like that, we need them as tools to really be able to like predict or like early on what therapies are going to be effective.
So it's sort of interesting how this is challenging. Overall, theranostic field is challenging because it spans so many different specialties. And in nuclear medicine, we're not new to saying, sort of device, detector, scanner type technologies have to play together with all of this radiochemistry and the production of those radiopharmaceuticals in the first place. But this is a whole nother dimension now, right? We get to therapy, we have to bring that radiobiology perspective in to be part of the field.
And if you think, I mean, like, let's like think about it a little bit, right? So if you think that maybe some of the chemists are not very well trained in physics, I mean, if you go to the biologists, I think that's even more less. They get even less training. So it's a lot of like learning on the job. So we need definitely expand the workforce in this area. So because in the end it's all about critical mass. So if you have enough smart people like working on the problem, that problem will like.
Yeah, but we do need focused problem statements, right? So
And we need to identify the problem.
Yes.
That is, and we need to understand. And like I always tell my grad students, it's like, okay, spend most of your time you should be spent thinking about the problem. And then once you understand the problem well, the solution will essentially present itself. Sometimes.
So I just have one last question for you, which might not be relevant at all, but so when you're thinking like a radiochemist in the lab with the flasks and the fluids, can AI help you? Is there a role for AI in self?
Well, believe it or not, I mean, chemistry was one of the first fields that used AI.
No.
With all this, screening libraries and things like that. Obviously, it's not very, popular, not popular, but very, it's not common knowledge, but the chemists have been using AI or some sort of machine learning type for computational chemistry for a long time. So all this screening libraries and things like that, modeling like interactions, that's optimization.
Yeah.
Those are optimization problems. Which
Yeah.
In the end, that's kind of like what AI is.
Yes. Great. Interesting.
From the master, I'm sure I'm gonna be pissing a few people. But I mean, that's how I see. I mean, if you look at under the hood, I think AI is just an optimization problem.
Yeah. This has been fun. Thanks for joining Rainier and me today for this awesome conversation. Let's talk again soon.
Looking forward to it.